Hello and welcome to the second lecture in this week of a discussion of dairy cattle and milk production. Laura Hernandez is an associate professor in dairy science here. Her interest is in lactation and its control of the development of lacteal tissue and the regulation of milk release, milk production. So I think we'll find that she has a lot to say to overlap with the talk we heard here last week.
Thank you for having me and for the opportunity to come talk about what I've been doing. It was an honor that Bob Brumwell thought I should follow him. If you come to my lab, you can still find stuff that belongs to him that has his name on it. They're like, that Bob Brumwell? So I kind of set this up that shows an evolution of how a scientist arrives at the University of Wisconsin and does something and becomes an expert in that area. I guess I'm an expert, maybe. But I work on serotonin and its function in relation to lactation and the physiology of the mom, and how things that are made in the mammary gland talk to the mom and allow her to make milk and also stay alive simultaneously. So this is a little story on how I became a lactation physiologist. It's not something I think you dream of as a kid. I'm going to work on mammary glands, but here I am. Here I was last year. My grad students always find these lactation rooms in airports or wherever, and they make me go stand in front of them and they take pictures, and I get really excited when I see them, as you can see there. But I started off as a little kid in El Paso, Texas, grew up in the city. My parents came from farm type areas, wanted nothing to do with farms, and I had an obsession with animals. So that was sort of how I got started. And this is just the Texas desert skyline that I miss, particularly on days like today when it's freezing. But the thing that I'm going to kind of talk about that is threaded through this is mentorship, and the mentorship that I've received that has really driven me to be where I am today. And this is my PhD advisor, Dr. Bob Collier, who actually worked for Monsanto and was the director of dairy business when they made Posilac. So he was the guy at the Senate on 60 Minutes that they were beating up as they went through that process. And he was my PhD advisor. This was last summer, 3 generations of what Bob likes to call lactation biologists. Dale Bauman is one of 2 animal scientists that's in the National Academy of Sciences, and he was my advisor's mentor. And so I was very much honored by a prestigious award at our meeting, and it was really fun to celebrate that with them. And they always said that your biggest success is what your students do, it's not what you do. They're the direct reflection, and that's how I try to run my lab. So mentorship is key, and I've had some really great ones. So how would one try serotonin and milk together? It was kind of by accident, as you'll find most things in science are. I was talking with Dr. Milo Wiltbank today, and we're convinced that anything that's been good has been an accident. It's not what you thought was going to happen. In fact, it's probably the last thing that you thought was going to happen. So when you're rejecting your null hypothesis, you're doing good things. So when I was in grad school, I walked in and Bob handed me this paper, and it was published in Developmental Cell, and he's like, read it, memorize it, come back to me and tell me what you want to do. All right, that's a big task. Developmental Cell, as many of you know, is a pretty high-ranking journal, very inundated with details and molecular biology. But this paper demonstrated that the mammary gland was a source of serotonin, non-neuronal serotonin, and that this was at least in an interaction with prolactin. And this was out of Nelson Horstman's lab at the University of Cincinnati. And Bob wanted to see if there was a way we could take this and link it into milk production or milk synthesis, and how really the mammary gland could regulate the cow and her ability to make milk, and what the coordination was with the mammary gland and the mom. And he'd worked on this quite a bit in tangential progress with his environmental stress work, but he really had an interest in this autocrine-paracrine regulation of lactation. So how does the baby keep driving this production regardless of what the mom's doing? So what did I learn first? This biochemical pathway. I wasn't going to go see him again until it was ingrained in my mind. So serotonin is basically a modified tryptophan And it's produced in this rate-limiting step through L-tryptophan to 5-hydroxy-L-tryptophan, which will become important and is where my research has been focused on since I've been here. And then in a non-specific reaction, 5-HTP is converted to serotonin. And then if you're in the pineal gland, serotonin is made into that hormone called melatonin that regulates largely your sleep-wake cycle and circadian rhythms. But what we know is that serotonin can be made in neurons, but it can also be made outside of neurons. And actually, what's made outside of the brain is what came first. So these peripheral effects of serotonin came way beyond the neuronal record in the story of evolution. So serotonin was first made in plants. And so really the focus of my research has been on the non-neuronal effects. And we've been working on utilizing this compound here, particularly to improve calcium metabolism in the dairy cow. And we're also looking at this in relation to human lactation as well. And I'll get into that in a second. So you have to build your toolbox. When you get to be a scientist, you want to acquire as many tools as you can. One of the things that I learned with Bob that has been very fortunate is how to isolate primary bovine mammary epithelial cells from cows. There aren't very good mammary epithelial cell culture that are transformed cell lines for cows. And this skill set is not widely available. So this made me a hot commodity for postdoc because I could do this. It's a really big pain in the butt, I must not lie. It takes about 18 hours and you're just sitting there mincing tissue, but it was a really, really great tool set. And I learned how to do it well and how to set up the cultures to ask the questions in the appropriate way. And that's what the science is about. What question are you asking? Is your model applicable? And how do you interpret your model to mean something real, right? Because a cell is a cell, a mouse is not a cow, a human is not a mouse, and we have to be able to put the pieces together and take the information away. So this model was really nice because we can put them in a three-dimensional culture system, so we embed them in collagen gels. And rather than just growing on a flat layer of plastic, they're able to actually branch and form lumens like you would see in a real mammary gland. And so we could actually test things that were related to lactogenesis and get answers that way. And so this was one of the first things I learned as a PhD student, and I spent a lot of time doing this. The other thing that I learned was how to milk a cow. Never thought I would have to do that. Never thought I would ever in a million years be milking a cow. I would call my grandmother when I was doing this and she'd be like, can you bring me milk home and we can make asadero cheese? That's a favorite back home. And it wasn't something I ever thought about. But it was an important skill that led to what you'll see in a second, which is milking a mouse. And I'll show you the video. But this is a half udder milking machine. We're using one right now at the Dairy Cattle Center. It looks like a big octopus, and we can measure milk yield in each quarter of a cow because each part is a separate gland. And normally the milk yield you get is all 4 quarters together. And so we're testing things that are neat about the mammary gland is you can test things at the quarter level and keep the endocrine status all the same, but manipulate what happens at the quarter because those quarters respond differently. Never thought I would use something like this ever again, and actually our current herd manager was in grad school with me, and when I told her I was going to do this, she really was not pleased, given the nightmare of this. But it was a tool, and it was something that I kept here in my toolbox that I could use later, and we're using it as we speak right now. My grad students are milking. So the next thing that we did when I was in grad school was you formulate a hypothesis, you want to do something new, you want to take risks, and by the time you leave, you're supposed to be the scientist, right? You're supposed to have these independent ideas and go off and have done all this cool stuff. And you kind of do, maybe, kind of don't. But I got to do a lot of really neat stuff that had never been done before, and a lot of it was my own ideas. And that was one of the great things about my mentor was he kind of gave me a little bit and said, design what you want. What are the questions you want to ask? And I think that's really important as a grad student. I learned how to fail. I learned that it was okay to fail. I learned that negative data was fine. It's totally fine. I learned that new things were fine and that people don't accept new things. And so experimental design is important. Statistics are important. So we described the bovine serotonergic system. That was the basis of my thesis. I learned a lot of cool molecular techniques. I worked with cows. I worked in cell culture. I did a lot of molecular biology, and I had a really interesting skill set when I left my graduate program. But most importantly was I had a lot of great friends that are still my friends today because we were all together failing over and over again. And this young woman here is actually our herd manager now. So they all come back. These are people at some point that you're going to run into in your career no matter what direction you go, and they're important. And those relationships I felt like were really important in my growth. So after that, Bob convinced me it'd be a good idea to do a postdoc. And I was like, oh, postdoc? I'm not going to make any money still. My husband was like, when are you going to get a real job? You're just learning. Learning's fun. So I went off to Cincinnati, Ohio, and I worked with Dr. Nelson Horstman, who originally discovered that serotonin was made in the mammary gland. This was like a complete 360 from what I've been doing. I was working with mice. We were doing breast cancer stuff. I was working with patients in the hospital, at the children's hospital, and really taking this to a human level. But I was a lactation physiologist, and the thing that we all have in common is that's why mammals are mammals. They lactate. And you can apply what you know in a cow to really help a human and vice versa. And that's what I learned during the 3 years I spent there. And this here is a video of my first grad student, Jimena Laporta, who would kill me if she knew I was showing this. And we were milking rats. I built a milking machine for rodents. We have it in my lab if you ever want to come see it. But I learned how to put things together and use what was there to ask questions. So we were able to do a lot of things in rodents that led us to a lot of answers that have turned up for the cows and have moved my research forward. But I was fortunate to do that while I was in Ohio. And you can see you can get a good amount from a rat, even from a mouse when she's at peak lactation. So these were my people when I was in Cincinnati. I worked with Dr. Horstman and his wife, Dr. Karen Gregerson, who was a neuroendocrinologist. I started looking at behavior, how antidepressants, SSRIs influence lactation, how to measure behavior, how to look at the molecular biology, how normal lactation physiology actually parallels what happens in the abnormal when you have breast cancer metastasis, and how to look at those things and try to piece it together to answer questions. And that's really what I felt like I got out of being there. So we did a couple different things. We were using an SSRI, specifically in this instant Prozac, and what we were able to show was that it affected the tight junction complexes in the mammary epithelium. So the mammary gland, like the gut, forms a blood-milk barrier, in this case instead of a blood-brain barrier or a blood-lumen barrier. And when you're lactating, you don't have things going back and forth because you're making milk and you're keeping blood here. And what we showed was that at high concentrations of serotonin or during the involution phase of the mammary gland as it would build up, it would cause the tight junctions that held that barrier together to fall apart. And that would lead the gland to have involution. And so we did this through some confocal imaging in our cell culture models, and we also demonstrated this in a cow model as well as in a rodent model. Using SSRIs. And that's one of the things that we've shown serotonin does, but it does a multitude of things. So then I got to thinking, what was I going to do next? I needed to put my big idea together and hopefully one day leave my postdoc and, as my husband said, get a real job and not just be playing around in the lab. And so there was a professor from Columbia University who came and gave a talk, and he was talking about bone and calcium and serotonin. And I got to thinking, calcium changes a lot during lactation. It's the major mineral that changes in the mom. And what we know in cows is that you'll lose about 80 milligrams per kilogram of calcium to the fetus. But what's so overwhelming is that during early lactation, you'll lose 500 milligrams per kilogram of body weight corrected to the metabolic size if you're making milk. So that milk production pulls way more calcium than it would be to support the fetus. And this can cause a problem in moms. And actually, it's a natural adaptation that mammals have evolved to lactate. And so what happens is you're going to deplete this blood pool, And the normal homeostasis that keeps calcium in check, parathyroid hormone, calcitonin, vitamin D, it can't quite keep up because this is not a normal situation. You're sending all this calcium to milk. Mom's got to keep her muscles going and standing, right? So they have to rely on a secondary or a different adaptive mechanism to support the lactation as well as keep the mom alive. And they do that through something called parathyroid hormone-related protein that I'll talk about in a second. So actually, what will happen is that you'll see a great increase in bone resorption during lactation. And a lactating mother who's exclusively breastfeeding for 6 months will lose up to 10% of her bone mass because she simply cannot eat enough calcium to keep up with the demand of the milk as well as her body. And the thought process was that after lactation ended, everything goes back to normal, no problem. But there's some recent data that's been coming out from different epidemiological sections of the world that suggests that this actually may not be true. And this is where we're looking at things right now. So cows are going to lose their calcium much more quickly than other species because they're dairy cows. They're producing way more milk than they would just to support their baby. And so they're going to be replenishing their pools 2 to 3 times a day. So they're losing a lot of calcium to support milk, more so than a lactating human would. So they're particularly challenged. So this brought me back to something Bob always told me when I was in grad school. Think like a lactation physiologist. That's what I am. Everything comes back to lactation. I'm a lactation nerd. I could think about it all day, every day, do it all day, every day, and I'd be perfectly happy. I skip all those meetings that they make me go to and just read papers about lactation. And so I thought, how could serotonin tie into this, given I'd seen that talk? So I was playing around in the lab and I was trying to bridge these things together. And I came across this hormone, parathyroid hormone-related protein. And Andrew Stewart, who was a clinician, and one of his fellows, John Weissel-Mersky, who's a clinician at Yale but also a scientist, had found this PTHRP in a malignant cancer that had gone to bone. And so it binds to the same receptor as parathyroid hormone, but it's not made in the parathyroid gland. It's made in all tissues, but it's secreted in times when you need something else. So the tissue can act as an accessory parathyroid, which becomes important for lactation. So it's closely related to PTH. They work through the same receptor. They stimulate bone resorption, but it's not regulated by vitamin D. It's coming from a different tissue to stimulate a factor. And the only time you can detect it in the circulation is during a metastatic bone cancer or lactation. And so the mammary gland has evolved this to take over to compensate for that loss of calcium. And Dr. Weissel-Mersky's studies, he's done over the last 30 years very elegant mouse studies showing exactly how it works, its necessity. It's also very important during embryonic bone formation as well. So there hadn't been much done with this in terms of the cows, and we'll see that in a second, but no one has really showed what had stimulated PTHRP. And after I had seen that presentation about serotonin in bone, I got to thinking maybe this is it. I don't know. So we took mice that were deficient in the rate-limiting enzyme to make peripheral serotonin, so tryptophan hydroxylase 1, which I showed you before. And what we did was we took mammary glands from them on day 10 of lactation, and this is a normal wild-type mouse, and this is one of our knockout mice. And this green staining is for the protein PTHRP. Lots of it present. It gets secreted into milk. In fact, it's 10,000 times higher in concentration in milk than in the blood. And you can see that there's very little in our knockout mouse. And when we injected these mice with 5-HTP, which bypasses that TPH1, we were able to increase the PTHRP concentrations. We also tested this in cell culture, so we used mouse lactogenic cell culture, and we saw when we added serotonin, PTHRP transcript was up, and this held true in our bovine cultures as well. So it appeared at least to be conserved within these species. The other thing that we did discover while I was a postdoc, but as you can see, sometimes science gets delayed. We just published this last year. We created a mouse model in which we selectively knocked out TPH1 in the mammary gland only during lactation. So the last 2 days of pregnancy and only lactation using a Krelok system. And what we saw was that when we knocked TPH1 out of the mammary gland There was no serotonin production in the tissue. But what was even more interesting was that we saw a 50% decrease in the concentration of circulating serotonin during lactation. And this is a wild-type mouse, and here would be those TPH1 deficient mammary glands, which tells us that at least during lactation, the mammary gland is supporting some of that circulating serotonin, which is why humans who are lactating, they make more serotonin. When they measure them in rodents or in humans, they tend to be happier. It's part of this probably mom-baby bonding thing, but it looks like the mammary gland is part of that response. And so this has been a really exciting part of the story. So we really believe that during lactation, the mammary gland is critical for this. So I finally got a job. I showed up here in 2011. For some reason, my department chair thought it was a good idea to hire me. And I took this piece of information with me. You come to start a lab and you're trying to figure out what your dogma is and what you're going to do and how you're going to coordinate everything and how you're going to do it differently and how it's going to be great. And the one thing that I really took with me that Bob always said and still to this day does is that the students you mentor and the people you collaborate with are a reflection of you. And their success is key to your success. And he said, the happiest day for me was when you got the job at Wisconsin. I did something right. And to me, that was a huge compliment because I don't think that highly of myself, but it really is. Like, my first PhD student got hired at the University of Florida straight out of grad school. And that, like, I cried that day. And I told my husband it was like more exciting than the day we got married, which was probably not the right thing to say, but I was really happy for her. And she's doing great. But these have been my students. So I got here, this is Jimena. I'm on a floor with Dr. Milo Wiltbank, who invented ObSync, the reproductive program that everyone uses across everywhere. I was lucky to end up on a floor with someone that is that crazy and that brilliant, and we have a lot of fun. But I also have amazing students. And when you start off as a young faculty, it's as much of a risk for you to take a student as it is for them to pick you. Because they're picking you over someone who has a lot of funding or is well established or is a big deal and has all these people out hired somewhere and doing great things. And you're just a nobody with a lot of ideas. And I've been really, really fortunate in that. And this is my daughter. I drag her along. My son will be the next one coming. He's only 9 months, but she likes to come and hang out with the cows or work in the lab already. But I have really great students. And so they have to be willing to take part of the crazy. And they have been. So we've done, hopefully you'll think, some really cool things since I've been here. So when I arrived here, one of the things that was in my mind, having been trained by Bob, is that not only do I want to do great science, but I want to do something that's relevant. Am I going to make a difference anywhere? I hope so. I haven't yet, at least I don't think so, but I hope I will. And so my thought was, if this was really going to have an impact on the dairy industry, I'd have to be able to feed the animals. It's a really easy way to do it. You're not giving injections. You're not manipulating them. And the way things are going, we want to kind of go this natural route. So the problem with that is that cows are ruminants. So they're a little tricky when you feed them. You don't always know if what you feed them is going to get out or not. So I had to start with the rats. So knowing how to work with the rats and the mice has come in handy for a lot of reasons. So Jimena got here and I said, we're going to feed all these rats 5-HTP and see if we can change their calcium metabolism. And she was like, all right, I'm going to milk rats. Sounds good. That was that video I showed you. So we had our little milking herd of rats, and we supplemented their diets with 5-HTP during about day 13 of pregnancy to day 10 of lactation to see if we would change calcium metabolism. And lo and behold, we did. We saw an increase in serum calcium on day 1 of lactation, and then we saw an increase in milk calcium by day 9 of lactation as a result of the 5-HTP stimulation. So we were seeing a response. We didn't have the details worked out, but this at least said, okay, if we can really make this work, it's worth trying to chase. So as Bob would say, in short, go big or go home. And that's kind of been the summary of my career. Milo and I were talking about that earlier today because we're meeting with the grad students tomorrow, and he's like, sometimes you just have to take a risk. And he goes, I've probably taken too many. And I said, well, there was a time about 4 years in where I thought I was going to get fired because I had nothing and I had no grants and I was out of startup money, but it came together. So you have to believe in your ideas. You have to believe in the science and do good science and believe in what you're doing. And so we really went big or went home with this. The next thing that Jimena went on to do, I'm a firm believer in understanding how things work. A lot of times we do stuff and we feed cows or we feed horses or whatever, and you're like, milk yield increases. Okay, is that a good thing? What else did you change to have to cause that? And I really believe you have to understand what you're doing before you go and start doing things that are going to change milk yield. I want to know why, and that's the scientist in me. So we went back to our knockout mice, and we were looking at trying to prove this bone effect, because that's hard to do in a cow. It's very difficult to measure bone metabolism in a cow. And what we saw was that back to these knockout mice that we were using that were unable to make this peripheral serotonin, they didn't have as many osteocytes, and they were a lot smaller, so they weren't chewing up the bone tissue quite like the wild types were. And when we added that 5-HTP back, the osteocytes would appear, they'd get bigger, they'd be more numerous, and a lot of the bone markers at the transcript level as well as at the protein level were consistent with this. So then, as we were trying to figure out further this mechanism, we had some weird things happen that we couldn't explain. And one of those was that in the mammary gland, when we were manipulating the serotonergic system, we were seeing big changes in the sonic hedgehog gene. Hedgehog signaling is like a target of everything in the universe: cell proliferation, embryonic development. It's really complicated. I get confused every time I look at the mechanism myself. But we kept seeing these really robust changes consistently, and we did not know why. So we had these knockouts. Sonic hedgehog would go down. The readouts, patch and GLI1, which are indicators of sonic hedgehog signaling, very lowly expressed in these knockout mammary glands. And when we added the 5-HTP back, they went back to the equal level of at least the wild type, if not higher. So one day, and this is what's great about the University of Wisconsin, I was sitting at this lab meeting with my colleague Chad Bazina, and I kept telling him, I keep seeing this, I don't understand, what do you think? And he was like, let's see if it's methylation. And I was like, why would it be methylation, Chad? Serotonin doesn't have a methyl group. And he was big into methylation, and he still is. He's like, let's just see, we can run all these tests and check this out. And I was like, okay, I mean, why not? So what Jimena did then was we wanted to look into see, we had read some papers that there was a linkage between sonic hedgehog and production of PTHRP, and that's pretty well established, at least in embryonic development. And we'd seen some papers as well where this would get turned on quite a bit in breast cancer metastasis to bone. So this brought it back to that calcium story during lactation. Was some of this also happening in a normal physiology? So Jimena went and used a technique called methylated DNA immunoprecipitation, where you can target a particular transcriptional start site within a promoter region without having to do pyrosequencing of everything. But if you have an idea of the target, you can really try to look at it. And what she saw was that actually two things, that the sonic hedgehog gene has two transcriptional start sites, one outside of the exons in the normal 5' UTR region, and then there was one within the exon. And then she saw that serotonin affected usage of these to turn on sonic hedgehog when you wouldn't normally expect it to be on. So it was changing the ability of sonic hedgehog to induce transcription and lead to probably cell proliferation and stimulation of PTHRP. So we saw that in our mouse model, but we also confirmed it in mouse embryonic fibroblasts, which is an established model for looking at sonic hedgehog that Dr. Rob Lipinski in the vet school has worked with quite extensively. And then we saw that hold up within our mouse model. So how does a prostate biologist, a lactation physiologist, and a cleft palate scientist start working together? This is how. And that's the beauty of Wisconsin. You start talking to someone and there's some kind of Wisconsin idea that pops up as a result of it. And it's been a really awesome collaboration. So because of this, we have an epigenetic thought, and my research has diverged into two arms. So I do stuff with the cows, but I'm also doing stuff looking at humans. And so we have this epigenetic thought of what we think is happening with the serotonin. So we think serotonin is altering methylation, and we've shown this with Sonic Hedgehog, and I now have data that'll be published soon that it also affects the PTHRP promoter directly as well. And as a result of this, we get increased production of PTHRP by the mammary gland to increase bone turnover, which allows calcium to be liberated to come to the mammary gland to go into the milk, as well as support maternal homeostasis. But we think serotonin's working at both sites. And we have a couple different hypotheses of what this mechanism might be, and we're trying to flush that out right now in the lab. So how does this tie to humans? We're a drugged culture. The most widely prescribed drugs in the United States are antidepressants. We are highly drugged and highly depressed, apparently. And these drugs have a lot of secondary effects. And one of the big ones that they have are on bone. In children, men, women, after menopause, during menopause, all stages, you can find some relationship with decreased bone health, whether it's hip fracture or bone density or bone breaking. There's all kinds of epidemiological evidence for that. Now, that poses a problem because not only are we drugged, a lot of women that are pregnant and lactating are taking SSRIs. And in the clinic, the doctors don't want to take these people off their medication because they fear for the safety of the mom and the safety of the baby. So as we got to thinking, we were like, well, if these women are taking all these SSRIs, are they setting themselves up to have really bad bone health later in life when a woman already is predisposed due to the loss of estrogen? And with this epigenetic model, we think we might have an answer or a way to intervene during pregnancy and lactation that would allow mom to stay on her medication and hopefully protect herself from this long-term bone loss. But we had no evidence. It was just an idea in our head. Getting back to being a lactation physiologist and how we put these pieces talking together. So I like to put cartoons about SSRIs, but side effects, this is what we would wish, include weight loss, clear skin, a full head of hair, multiple job offers, abs of steel, and a sparkling personality. But really, the side effects are not great. We laugh, but there's a long list of things that can happen long term. So I mentioned that recently there's been a couple of epidemiological reports that have come out and said that irrespective of vitamin D status, diet, where you live, smoking, alcohol, all these things that could influence bone health, time spent breastfeeding, cumulative months breastfeeding, and I'm not talking how many times you've been pregnant, that was also a part of the model, the statistical model. Time spent breastfeeding has increases the risk of a decreased lumbar spine bone mineral density as you go forward in life, all by itself, with nothing else. And we are pushing people because we know breast is best, but sometimes it doesn't work out. Sometimes people don't want to, sometimes they can't, and all of those things are fine. But breastfeeding is hard, and you layer pushing this with taking medication because you're depressed or other things. What could we be doing to mom's bone health? And for a long time it was thought that you would breastfeed, then you'd stop, and then your bone would come back and you'd be fine. But there's been several reports in different parts of the world that are showing that this holds up, and it might be a bigger problem than we originally thought. So in the United States, we've recently come up with the Healthy People 2020 objectives, and our goal was to increase the number of people who've ever breastfed. We really would like to get up to this exclusivity at 6 months. Can you breastfeed exclusively for 6 months because of all the health benefits it conveys to the baby? And so there's a whole bunch of goals that we're trying to hit, and I was at a meeting in November that said we've actually hit quite a few of them already, and we're only in at that point 2017, but We're doing better, but we're pushing this. But we don't know a lot about how to support mom. We don't give mom a lot of support. She has her baby and we say, good luck, see you in 6 weeks. You go take the baby to the pediatrician, and the pediatrician's like, you doing all right? I hope so. If not, I can send you to a shrink if you're feeling like you're gonna harm yourself. And so that's a whole nother set of social issues that we have with this, besides the physiological events. So we got to thinking, we were wondering, if you increase the serotonin by taking these SSRIs during this pregnant and lactating time frame, are you predisposing yourself, setting yourself up for failure once you hit later life and you're already going to have a problem with bone as a woman because of the estrogen? So the first grant review we got back from NIH was, is this even a phenotype? You have to prove it to me. And there's no data in the human. We could not find data anywhere, mining everything. So we took mice, and I should have put the picture in here because we even have them out to 9 months, but we took mice and we gave them Prozac through their whole pregnancy and lactation. And then we let them sit there. We stopped giving them the drug, and then they sat there for 3 months and 9 months after we weaned them, and with just one lactation to start. And what we saw was that the bone volume per trabecular volume measured through micro-CT was decreased at 3 months and even further decreased at 9 months compared to the wild-type controls. So this shows that, at least in this mouse model, you do get a decrease in bone volume. And so that was a really exciting proof of concept for us. And my program officer says he thinks we're going to get funded, hopefully this time around. But we really had to try to figure out what's going on. And I mentioned that methylation mechanism, and we think we might be able to reverse this by giving folic acid, high-dose folic acid, or maybe even choline or methionine as an alternative. So we're looking at ways that we can help mom so she can stay on her medication, support her to lactate, and then hope that she's not going to have osteoporosis as a result of lactating. So what does this have to do with calcium in dairy cows? I'm in the dairy science department, right? So there's a lot of things I had to establish to show that this was a real thing. I mentioned in science, you're doing new things and you have to prove to people that they're real or that they exist. And there was lots of literature about this in rodents and humans, but rodents and humans aren't cows. Cows are cows, right? So one of the things that we had to first do is we went out on a couple of different dairy farms. This is my former grad student, Spencer Moore. And he took blood samples every day from -5 before birth through day 10 of lactation, and then he had 5 more time points out later. So for 4 months, every single day, he was out on a farm not far from here, 2 different farms, taking these blood samples. And we needed to know, could you measure serotonin in a cow? What was it? What's normal? I don't know. I had no idea. And that was one of the first things we had to do. So this red graph here is serotonin and the green is total calcium. The second thing we wanted to establish was, were we seeing this same relationship with serotonin and calcium that we saw in rodents? And what we see here is that, and we're not sure yet what drives the one or the other, but we see that as serotonin decreases around parturition, so does calcium. But we don't know which one is first and how they're intricately tied. But we saw this on this farm. We saw it on the second farm, which was completely managed very differently, much more aggressive management style, different breed, but they do everything to make sure their cows are healthy, and you can see their calcium concentrations don't change as much at parturition, but the serotonin followed the same pattern. So this at least gave us some inkling that what we were seeing in rodents and what we had hypothesized might hold up in cows, but we had to do this. And let me tell you, as a scientist, no government agency funds this. This is why you go to a university and you hope that you make the best of every penny that your startup package comes with. Because this is, you don't know what's going to happen. And that's not what the government wants to fund. They want to fund what they know is going to happen. So these are big ideas you're trying to deal with when you start up, to convince people that you know what you're doing. So after we'd done that and we figured out that we could give 5-HTP and manipulate the calcium metabolism in the rodents, we wanted to see if we could do it in the cows. But we had to give it to them IV, because we were unsure of what the rumen would do, because the rumen has all these bugs in it and they can degrade things and change them, and we have no idea, at least at this point in time, we didn't know what was going to happen. So these cows got 3 different concentrations of 5-HTP intravenously in their jugular veins. They were given 4 doses over a course of 4 days, and we did a time series measurement to see how these hormones and calcium metabolites were responding to a direct stimulation of 5-HTP. Everything we had done prior to this, we'd measured like 10, 20 days after we'd started our treatment. So we were taking samples at 5, 10, 30, 60, 90, and 120 minutes after we gave our dose. And I'll never forget this day. It was a great teaching moment for me and for Jimena. She put this graph together and she came into my office and she was finishing up her dissertation and she's like, I'm gonna fail. We've been lying. Calcium went down. It's down. It went down. We lied. Everything we did is wrong. And that is where she was at. She was almost in tears and she was like, what are we gonna do? Everything's wrong. We're gonna have to retract all our papers. And I was like, Jimena, let's sit down and think about this for a second. So we had never looked at it calcium in direct response to the 5-HTP. We looked at it 13 days after we've been giving treatments. But calcium is modulated through classic negative feedback. If any of you have taken endocrinology, you know things go up, or biochemistry, something else goes down, right? It's this negative feedback loop that regulates biology. And so what happens, and as we've built the pieces, it's really leading towards this, and I didn't put some of the mouse data in here, is that serotonin, at least one of the things it does is it drives calcium into the mammary gland. And fortunately, we'd taken urine and milk samples. So we took urine samples before we gave any treatments, and we took them again at the 120 minutes after the treatment. And what we saw was actually that there was a decrease in urine calcium output with the higher doses. So the cows weren't just excreting it into the urine. And because these were late lactation Holstein cows, we also had milk calcium available as well, and we saw that these higher concentrations, we saw an increase of calcium into the milk. I suspect there was probably more gut uptake of calcium as well, but that's a little trickier to measure in a dairy cow, so we don't have that information. But it wasn't that they were being excreted into the urine. The cow was using the calcium. And this had been consistent with some of the things we'd seen with the different pumps and channels that move calcium into the mammary gland and sequester it and help secrete it into milk. So she sat down, she went back to her office, she kind of poured through it, and then she was like, okay, it's fine. But it was a great teaching moment. We never think what's gonna happen happens. I mean, this was not what our hypothesis was, right? But it's really important to think about the biology. So take a breath, slow down, and think about the biology. Yet another part of wisdom that Bob would give me on a daily basis as I was running up and down the hallway doing many things at once, and I still do to this day. So then we went on to the transition cow. So that model before was a late lactation dairy cow. That's not the cow that we're really concerned about. The one that's about to give birth and make all this milk, where she has problems because she's going to have to start giving 100, 150 pounds of milk and keep her metabolism up, and that's where they're really susceptible to problems, and that's what we're trying to help. So my current PhD student, Samantha Weaver, she did a transition cow study, so we started giving the 5-HTP to the cows about day 8 prior to parturition. You can only estimate that so well. They are animals. They will give birth when they feel like it. And the blue line are the control cows and the red line are the 5-HTP treated cows. And what she saw was that the total calcium concentrations were increased in the animals that were given the 5-HTP prepartum. So this looks like we can give this to the cows, hopefully eventually in their diet, in their nice pens, cushy pens as they're hanging out, getting ready to give birth. And that's the end goal. This is still IV because we're working out the logistics of that, but this corroborated what we were seeing in the rats. So we were on the right track, at least, hopefully. This was then confirmed. My collaborator in Switzerland, he did a similar study in their model system where they graze. So their Their dairy housing is a lot different than ours. They don't have these conventional barns where they're getting these very fixed, beautiful rations that we fix to give them the exact food that they need. They get a little bit of that, but they're also out eating grass and milling around, so the system's a little different. They also don't produce as much milk as our cows do either, but they still produce quite a bit. So what he did, so he gave the 5-HTP similar to us about 8 days prior to parturition. They saw that serotonin increased with the 5-HTP treatment, which we would expect it to. But what he saw was a replicate of what we saw, that the cows with the 5-HTP had higher total calcium concentrations around parturition. But what he did for us was confirm something that we'd kind of been arguing about. PTHRP versus PTH, the classic parathyroid hormone versus parathyroid hormone-related protein. So what he saw was that in the cows that did not receive 5-HTP, so they received saline controls, they had a spike in PTH, but their total calcium concentrations were lower. And the cows that were receiving the 5-HTP didn't have a change in PTH, and they had higher calcium concentrations. And that fit with what we were seeing with the PTHRP. The other thing you'll learn about science is that to run all these assays is very expensive. And you wish you could run all of them. And sometimes it's just not possible. So you have to pick and choose and plan, and that's why collaboration is really important, to fill in the gaps that you're missing. And so this piece of information was really, really important to our story as far as really thinking that serotonin works with PTHRP and not PTH during lactation. A second collaboration I formed was with a researcher at the University of Florida, Dr. Jose Santos. And one of the methods we use to control calcium in dairy cows is by changing their anionic status prior to birth. So what they found was that if you gave them salts that would make their blood a little bit more acidic or create a metabolic acidosis, so to speak, you would get better release of calcium from the bone when the animals gave birth. And this was done over like 40 years of research. The products are much better now, but it's something that people use in the dairy industry. So I was getting questions. Negative DCAD diet versus positive DCAD diet, so you're acidotic or you're not, does that affect serotonin? I didn't think it would, but it was a question I kept getting. And so Dr. Santos was performing a study down at his university in Florida, and they sent me some blood samples, and what we saw was that these acidified cows prior to birth had higher serotonin concentrations, and when we stopped acidifying them at parturition, you can see the serotonin levels return to normal. And this was completely blew my socks off. I mean, I had no idea why. I'm still not sure why. But it looked like this could be a contributor to the mechanism of how this worked. So we went on to do a study here with one of my master's students, and again, we were doing this acidified diet, and we were also giving them 5-HTP. So we wanted to see, do the two things work together better than either of them alone? So if we're actually going to use the 5-HTP as a way to help cows, is it enough by itself? Or if you do it with something that's currently being used, is it even better? Does it compete? We need to know these things, right? We need to know how it works. So what we found was, and this is ionized calcium concentrations, this purple line are these cows in the metabolic acidosis before birth and also receiving our 5-HTP compound for that week prior to giving birth. Their ionized calcium concentrations were the highest. And the acidification alone and the 5-HTP alone were about similar. They were about even. And cows that are negative control receiving neither of them were the worst. This held true also with total calcium concentrations. The combination of the two things together, we had the best response. Each treatment alone, so acidosis versus 5-HTP, were about the same for total calcium, and nothing was the worst. So that was pretty interesting as well, and at least supported that if you're going to maybe use both of them, it might work better, but we need to understand what they're actually doing together. So what we didn't find that they found in the study in Florida was that the negative DCAD diet didn't change serotonin concentrations. We see our responses when we give 5-HTP, the serotonin goes up, but when we just acidified the cows like they did in Florida, we didn't see the change in serotonin. Now, those cows have sunlight, they put weird things in their diet like citrus pulp and other things, and so we can't take into account that maybe something they were fed was different from what we were fed, feeding our cows that could cause that. But we're going to have to look into that further in future studies. So we weren't able to see that same exact effect that they did. But we did see a synergistic effect on calcium with the acidosis plus the 5-HTP. One of the things that we did see in this study was that there was a change in blood pH, which you would expect because that's part of what's happening. So the theory is that you make the cows a little bit metabolically acidotic and their PTH receptors become more responsive to the hormones that allow them to mobilize calcium from their bone tissue. And the negative DCAT diet with the 5-HTP together, their blood pH was lower than any of the other treatment groups. So something about the acidosis plus the combination of the 5-HTP was causing this. I can't find anything in PubMed that would tell me why. I have no clue why adding this modified tryptophan along with a bunch of anions would do that, and it's something that we're trying to pursue further in our upcoming studies. But this is kind of some of our new data. So then we got to the point, what's the cart or the horse? I showed you guys those graphs where serotonin goes down, calcium goes down. Does one stimulate one or the other? Do they stimulate each other at all? Are they independent? Do they overlap? We don't know. So if we have this total blood calcium pool we're trying to maintain, there's a couple things that could be going on. We have an increase in serotonin that occurs when you get outside of this range. And that's driven by, in the case of lactation, pushing calcium into the mammary gland for milk. And we see increases in these pumps when we give serotonin in the mammary gland. So we're getting more transport of calcium. Then we increase our milk calcium. And this drives a decrease in blood calcium concentrations that lead to this bone turnover. That's one route. So the serotonin goes up and causes this cascade. Alternatively, you get a drop in blood calcium that occurs due to the movement of calcium into the mammary gland, and that stimulates production of serotonin by the mammary gland, which drives this PTHRP bone turnover response. It could be either one of these, or it could be both. And we're trying to tease out what's the driving force. So we did a study in which we fed cows 3 different levels of calcium. So one was a low calcium of about 0.4% of the diet, a medium level of calcium about 1%, and then our high level of calcium was 2% of the diet. And they got these diets for about 21 days, and then we challenged the cows with EGTA, which is a selective calcium ion chelator. So if you put EGTA into a cow, or an animal, yourself, you're going to chelate your calcium. That's what's going to happen. It's going to bind it up. So what we did was, after they'd had those diets, we had challenge days, and we treated these cows at a constant rate. So they were set at a flow rate using basically like an IV pump, and it was set to flow at 500 mils per hour, and they were getting this EGTA solution, 5% EGTA. And we were measuring blood samples every 15 minutes after the start of the infusion. So we took a blood sample, and what we were trying to get to was, our endpoint was 60% of where they started from. And that's when we were going to turn it off and then let them recover. So we took blood samples every 15 minutes. And what we found was that the cows on this high calcium diet were more resistant to the challenge of the chelation. So this left side of the graph, they were slower at decreasing compared to the medium and the high calcium cows. Once we get to the right side of the graph, they were just overall higher, and the slopes of these lines are all the same. And you'll see in a second, on average, it took these high calcium cows about 450 minutes to get to that 60% of where they started from, but we can't make statistical analysis past 360 minutes because the other groups were losing cows, and we didn't have 3 animals to run statistics on. So this was really interesting, that they were resistant to the challenge, because our hypothesis had been that they would recover better. So you'd challenge them, and then you'd turn the EGTA off, and those high CALS would take off faster than the medium and the low. And they all recovered the same. Actually, I don't have the graph in here, but all 3 treatment groups, when you turned off the EGTA, they increased at the same rate back to their calcium level, which tells you how homeostatic calcium is. It's a really tightly regulated system. So they were resistant to the induction, but they all recovered at the same rate. We also measured serotonin at these time points. And what we found is summarized here. So this is the average of the low calcium, the medium calcium, and the high calcium CALS during that hypocalcemia challenge. And what we found was that there was a treatment effect, and the medium calcium CALS had the highest serotonin concentrations. The high calcium CALS were intermediate, and the low had the lowest serotonin concentrations. And what we know is that cows in this medium calcium range, they don't know if they're too low or too high. They're trying to figure out, do I need to turn on negative feedback or do I not? And this kind of made sense then. So serotonin's also up because they don't know where they are. Do they need more calcium? Do they have enough calcium? So we're turning on this other system to help us deal with our calcium problem. And we know that cows, prepartum, if you feed them a diet containing of about 1% calcium, they're at the highest risk for having periparturient hypocalcemia or milk fever than cows in the low or the high ranges because of the negative feedback loop or just the overwhelming of the system. So this was a piece of information that was really interesting to our story of trying to figure out what serotonin does. So state of the art, where are we today? What we think is happening is that calcium is being shuttled into the mammary gland. This is through a ORI1, which is a calcium channel. And then 60% of it gets pumped out through this plasma membrane calcium ATPase. 40% goes bound to the protein and is getting pumped into the lumen of the epithelium to make milk. This decreases calcium concentrations, and 2 things happen. There's a calcium sensing receptor on the mammary epithelium like there are in other tissues, and it tells you, is your blood calcium high or low? So we know that calcium interacts with this, but we also have shown that serotonin does as well. It stimulates expression of this at both the RNA and protein level. So we think this decrease in blood calcium stimulates this receptor, as does likely serotonin, to increase production of PTHRP. And John Weissel-Mirsky's work has really showed this classic pathway in the mammary gland. And then once we get more PTHRP, this goes to the circulation, stimulates bone resorption of calcium to compensate for the inability to take enough calcium in through the diet. And that would hopefully bring your calcium back to normal. We also know that serotonin is acting directly on calcium. We think it's shuttling it, stimulating it to be taken up into the tissues. And we also know that serotonin directly acts on PTHRP to drive increased production of the protein. So it appears that serotonin is working in 3 different places that we know of to date to drive this calcium homeostasis during lactation. And we're still trying to tease all those pieces out in both our cow story as well as our SSRI human bone health story. So with that, I have to thank, of course, funding. I've been very fortunate, and then I have all these amazing students that have worked with me from the undergrad to the graduate level. We have anywhere from 4 to 6 undergrads working in our lab at all times. Most of them have either ended up in vet school, medical school, or grad school. They've all gone on to do something much better than me and done great things, and I still am doing lots of fun stuff with my mentors, Bob and Nelson. But really, these guys make it happen. They're the ones out milking right now, and when I show up with an octopus-looking contraption and I'm like, I have a crazy idea, you should do this, and they're like, all right, sure, sounds good. I bring pizza and donuts usually to make up for it. But I've been really fortunate being here at Wisconsin, and I've had a lot of fun. So hopefully one day I'll be as great as Dr. Bremmel or Dr. Shook back there too. So you have to thank all the people that help you because they're really what make the lab go round. And if these guys aren't successful, I'm not going to be successful. So really, it's in my best interest to give them the best education I can. So I try. And with that, any questions I'm happy to take.
Is the presence of serotonin as part of the lactation process by human design, there by human design to encourage?
Part of it is probably some of that. But one thing about serotonin is it doesn't cross the blood-brain barrier. So if it's made in the brain, it stays in the brain, and if it's made outside of the brain, it stays outside of the brain. So it seems like it probably evolved to help metabolism first, but then once we got complicated and became humans, it also helped drive neuronal stimulation that drives the bonding. Yeah. Other questions?
I never thought about the problem of getting drugs into a cow through the lumen.
Mm-hmm.
Is there any drug that can be fed orally and works?
So what they typically do is they'll rumen protect it. So they use chemistry to keep it from getting degraded in the rumen, so it'll bypass, and then once it hits the abomasum, which is the true stomach, it'll work like normal, and the hydrochloric acid will allow it to become free and absorbed. We have done a study. This is like new brimming data. We did a study where we were sticking the 5-HTP straight into the rumen of the cows, and it looks like it gets absorbed. So we can increase blood concentrations at a pretty low dosing of 5-HTP straight into the rumen. So we may not even have to biochemically protect it.
No other questions? Thank you very much.
No problem. My pleasure.